So without further ado, I'm going to turn the microphone over to Dan. And again, Dan Smith is with Cobra. He's the Chief Scientific Officer. His presentation, Manufacturing for Chimeric Antigen Receptor, T-cell and Gene Therapies.
Thank you. Good afternoon, everyone. Can you hear me okay? Yeah, good. Right, so this afternoon's presentation is really about the need to produce critical starting materials for the generation of CAR T therapies. So I work at Cobra Biologics. What I want to do this afternoon is give you a quick introduction to Cobra, very brief introduction, then talk a little bit about CAR T therapies, giving you an overview and the manufacturing complexities that are involved in those types of products. Then the challenge to produce critical GMP starting materials and our solution to that, our solution being high quality plasmid DNA, and then a quick summary at the end. So moving on, Cobra Biologics. So this is our mission statement up here. I'd just like to say Cobra's been around for a number of years and has been very active in the area of gene therapies. It started off as a product company itself, delivering and manufacturing plasmid DNA for the use of gene therapy products. We very rapidly moved into the CMO market and now have a wide offering of protein, virus, DNA, fill finish of API, and also whole cell vaccines and biologics as well. Cobra is based in Europe. It's split across two companies. We have a site in the UK which is focused on microbial plasmid production and viral vectors for gene therapy. We have two sites in Sweden, one focused on mammalian antibody production and the other focused on large-scale commercial microbial production and fill finish capacity there as well. So, moving on to the main topic of the talk today, CAR T therapies. So the story so far, everyone's heard a lot of hype about CAR T, everyone's heard a lot of really, really good results that have come out. In fact, today, a company announced today that people that are treated on their clinical trial, they've seen 100% remission of those people on that trial using a CAR T therapy. So this started back in early 2011 when the first trial was performed. And actually, that was a very early demonstration that these CAR T cells can successfully treat patients in late-stage disease. There has now been a major trials going on all over the world in the areas of multiple oncology indications. It's a very new and exciting sector, and there's been a lot of activity, space scientifically, a lot in the corporate partnering area, and you can see on the table on the slides here actually there was some really big IPOs actually since in 2014 and 2015. Really with companies like Juno, Selectus, Kite Pharma leading the way in this area. Currently these types of products are really aimed at late-stage disease where there's no other modality there to treat anything. However, it is anticipated these type of therapies will replace the standard for cancer treatment in the future for a wide area of oncology indications. So what are they? What are we talking about when we talk about CAR T therapies? Well, CAR T therapies have a very complex biomanufacturing structure. Really what the companies are trying to do is take out the T cells from a patient, engineer those T cells with modified antigenic receptors, TCRs, T-cell receptors, and implant those back into the patients to derive an immunoresponse against the oncology indication that they're targeting. In order to do that, they need to transduce those T cells. And the way they transduce those T cells, in the majority of cases, is to use a viral vector, so a safe form of a virus that's been engineered to deliver a transgene of interest for the chimeric antigen receptor. So in order to do that, you need to make that viral vector to start with. So you can see it's quite a complex process to even get into a patient to start with. You need a lot of complex raw materials, starting materials to build that through. If we take that a stage further, we can look at complex supply chains in those areas. So right at the start, you'll start with a piece of plasmid DNA. The plasmid DNA for your therapeutic chimeric antigen receptor itself. And the way a lot of these viral vectors are made is by a triple transfection approach. So you need 3 plasmids, one to supply the helper functions to generate the virus, one to supply the viral genome itself and the structural proteins, and one to carry the transgene in. So all of these are critical starting materials. You then manufacture the viral vector of interest. That viral vector is then used to transduce those T cells that are removed from the patient. and then selected out for the ones of interest and engineered out, selected from them, and then reintroduced to that patient. So it's very complex. There's a lot of things to think about here. And the point I want to make here is actually that in the EU, and that's where we do a lot of these things, plasmid DNA for this type of indication is classified as a GMP starting material. So, very complex supply chains, a lot of things to think about, a lot of things to come together before you can actually get there. This is very critical for the discovery phase as well. What's the right molecule? How quickly can we move through this supply chain and get interesting viral vectors made to make interesting engineered T cells, which then go on to have the promise. So again, a lot of work to be done in the discovery development interface for these things. So what I wanted to focus on really was plasmid DNA supply for this, because that's right at the start, and you've got to get that important right at the beginning. So from a regulatory perspective, plasmid DNA is used in the manufacture of these products and is classified as a GMP starting material. So a lot of customers ask us to make plasmid DNA for these indications. And we have a variety of offerings and a variety of services to do that at Cobra. Some drug developers really want all of their plasmid made to full GMP grade manufacturing, and that's fine. But there does come with an increased time pressure and an increased cost pressure to be able to deliver that as quickly as possible for the customer. Others have sought perhaps a more timely and cost-effective approach to get their products into clinic. So as I said, plasmid DNA is classified as a starting material when used for these applications under EU clinical trials guidelines. This less stringent requirement of a critical starting material allows us to do some rapid and streamlined bioprocessing for the generation of this type of product. However, we still have to remember that we can't just make plasmid in a lab, at a lab bench, with no care and attention to what we're doing. It still falls under a GMP umbrella with regard to the manufacturing standards, the controls, and really the manufacturers, such as us, need to address these things in order to allow them to be used in further clinical application. So, why am I here today, and why is this of interest to us as Cobra? Well, like I say, we've had over 20 years experience in plasmid DNA production technologies. 16 years of that has been in the contract manufacturing environment. We've worked with over 29 customers globally for plasmids alone, with over 100 batches of plasmids made at our facility in the UK. 37 of these, well, it's greater than that now, have been full GMP grade batches of plasmids. So we know what we're doing, we're making GMP grade material. And interestingly, 23 batches now have been made through the high quality route that I wanted to talk about today. Well, other technologies there to support that, the use of antibiotic resistance free plasmids, so we don't have a chance of introducing the wrong transgene into the virus or misincorporating antibiotic resistance into that viral vector as well. We can offer full analytical support, cell banking, and there's lots of patents and publications to support all of this. But the real crux in all of this is actually how do we make a streamlined approach for high quality plasmid DNA? So what we've developed at Cobra over the last 18 months to 2 years is the solution for this in some ways. So it's fully traceable raw materials are still used in the manufacture of this. Traceable components are used in this. And we use a segregated production environment that meets regulatory standards throughout the production phases. We tightly manage those supply chains to allow reduced manufacturing times from project initiation to product release, allowing that product to go on to make viral vectors and then first-in-man studies for these types of products. We've established a highly flexible and responsive approach to this manufacturing, and this complements very nicely with our GMP service, and it allows our customers the real choice for phase-appropriate plasmid DNA manufacturing. So it's consummate to the phase that we're working in. Preclinical, early phase, a lot of customers are focused at high-quality DNA. At late phase and maybe commercial, they can move on and the process transfers very nicely into our GMP grade offering. And important in all of that, and what's important from the bioprocessing angle, is that the plasmid recovery and the purification steps share a common platform. So that allows the movement from high quality through to GMP grade seamlessly and very smoothly. So actually, why am I here talking about plasmid DNA anyway? I mean, I wanted to try and make it a little bit more interactive People in the audience, who's made plasmid here? How many people have worked at a bench in their time and made a bit of a plasmid through a kit? No one, brilliant, okay. But a lot of people do it in research labs. You can buy a kit, you can express them, you grow some bugs up, you transfer them, grow them up, get the plasmid out, there you've got some plasmid through the kit. Fine, so what's the problem? What's the big deal about this? And this is what a lot of our customers say, what's the big deal in this? So what we have to think and what we have to remember about this is that the plasmid DNA that we're making right at the early start here for these therapies carries the therapeutic template in its gene sequence to the product at the end of the day. So all errors that are introduced at this stage will be replicated and carried through to the end product. So although it might seem like a very early stage starting material, it's critical to get it right. Cell banks and DNA templates last for the product lifetime. They're not just for early phase studies. What you've got at the start will be carried through hopefully to your commercial manufacture at the end. So there's a real need to understand the long game here. So we have to get it right from the start. So how have we done this at Cobra? What's the solution for our HQ high quality plasmid offering? So what we've done is we've taken the understandings from our GMP process at large scale and streamlined that and scaled that down. So to start with, we use a GMP compatible cell line. So our E. coli cell, our DH1 strain, has been used for a number of years in this area. So we take that from a master cell bank at GMP grade and use that for the high quality side of things. We do a production and transformation and clone selection of this and make what we call a production cell bank. That production cell bank is QC tested and released in the typical way you'd imagine a cell bank to be released. So we have full sequence analysis done on the plasmid it's containing. We look at growth studies for that cell bank. We also look at bacteriophage testing of that cell bank to make sure it's right from the start. And this takes 2 weeks from start to end to do that. And I'll come on to some of the other analytical tests that we do on this to ensure quality is introduced right at the start for this. So 2 weeks from initiation through to that production cell bank and release of that cell bank through testing. We then move into the production phase, and this can consist of shake flask production, typically 12 to 24 shake flasks, or if needed, going into a 5-litre tank stirred reactor type approach. The plasmid is then made through that production phase and recovered by conventional alkaline lysis steps. Again, a scaled down version of what we use for our standard GMP process. We then subject it to a 2-stage purification strategy through ion exchange media and GE's Plasmid Select chromatography resin. It's then diafiltered into the buffer of interest for final formulation and then filled. And then analytical testing is performed of that plasmid under qualified methods that are identical to the GMP methods employed for the same large-scale prep for GMP manufacture. And more importantly, it is the target specification that we use for that is consistent with the GMP grade plasmid as well. And all of that takes 5 weeks to do there. So just thinking about the quality that we've put in here, because this is high quality plasmid DNA, it's not just a kit. What do we mean by that? Well, during that initial 2 weeks of cell bank selection, we're looking at clone selection as well. So we take 6 clones off the plate, we grow them up, we look at them, and we look for evidence of structural rearrangements of the plasmid. And there's 2 examples here. The left-hand side of the slide showing no evidence, and all of those clones could be selected for forward movement. The right-hand side of the slide shows that one of those clones, clones 3, actually has a multimeric form of the plasmid produced within it. So that clone's discarded. So right from the start, we're selecting clones that have the right product of interest to move forward. We then make the production cell bank and we test it for genetic stability. Although we're doing a small-scale grow and enough plasmid to get you through to viral vector production, we want to ensure that if that cell bank is used for forward manufacture down the line at any point, we know that it's stable enough to go to high generation numbers. So again, we look at generation numbers out to 60 cell generations and look for evidence of structural rearrangements or segregational issues. And again, sometimes you can see loss of plasmid, sometimes you can see structural rearrangements. So those ones are eliminated and we'd pick another part of the bank and go back and have a look again. But in general, we see a good degree of genetic stability in the production cell banks in exactly the same way as we would see it from a GMP side of things. So you make your production cell bank, it's tested, it's released, you've got it, it's ready to go. We move on to the actual production side of things, shake flask grows, and then plasmid DNA purification. And what I wanted to do in this last slide, this slide here, is just show you an example of this plasmid purification step. So we use a 2-column step purification, ion exchange chromatography, which is bulk separation of plasmid from host cell contaminants such as RNA and host cell protein. And the table at the bottom here shows the reduction level that we see across that step. It's still not what we want to see from the final material. Our bulk drug substance targets is lower, but we've still got another purification step there. We then move on to what we think is a really nice polishing step, and we've designed this with GE over a number of years. And this is their Plasmid Select chromatography media. And this really enriches for supercoiled plasmid. And the supercoiled plasmid is the form of the plasmid that you want for this high quality application. And you can see on the little gel at the bottom here that actually the load going on contains both open circular forms of the plasmid and supercoiled forms of the plasmid. We can elute the open circular off specifically, leaving the supercoiled on the column, which we can then recover and move forward into the diafiltration and the final formulation steps. And we see very, very nice enrichment of this plasmid across that time. So again, what are we looking at the end of the day here? So we're actually analysing this plasmid with the same methods that we'd use for GMP grade plasmid. And we can see that it actually hits those specifications time and time again. The key one here is the percentage supercoiled by CE analysis. Typically we see 95% levels of supercoiling. We've run 23 batches of this now, and I've just averaged 20 of them here. The other 3 are fine as well, I just did it for 20. 95% is the level we get there. So really, really good consistency and level of purity and supercoiled level in those plasmids. So the product is delivered to a high quality. It provides confidence for that forward manufacturing. So in summary, CAR-T therapies have a complex biomanufacturing supply chain. They require rapid and flexible access to critical GMP starting materials such as plasmid DNA. We believe our high-quality plasmid DNA offering gives you that confidence and quality of plasmid that you require to progress CAR-T therapies rapidly into clinic. It's a streamlined and scaled-down version of an established GMP plasmid DNA platform. It's a fast production process, 7 weeks for a new plasmid that comes in. And when you think about it, you're trying to make 3 plasmids for AAV production for viral vectors, or maybe 4 or 5 plasmids for lentiviral vectors. That's a lot of plasmids that need to be generated quickly. 5 weeks for repeat production. We can do a scale from 100 mgs to a gram of product in our production runs at this scale. So that's really nice, allows people to move through preclinical and early phase development very, very quickly. Fully traceable raw materials, traceable components, and segregated production areas play into this service. And the production of the cell banks and the plasmids undergo full analytical testing, ensuring that a high quality product is generated. And therefore, we believe this is a rapid and flexible approach to obtain plasmid DNA GMP starting materials. I'm going to stop there. Thank you very much for your kind attention. And I'm open to any questions if you have any. Nice.
I was looking for some insights on what happens in the T-cell expansion. Is that something you guys do? Is there any challenge there at all?
Yeah, it isn't something we do. We do work with customers that then go on to do that themselves. I mean, one of the main challenges in that T-cell expansion is actually selecting the clone of interest out of that pool, really. And a lot of people use different approaches for that. So they might actually manufacture ligands specific for the TCR of interest and then do some affinity chromatography to pull them out. They're looking at FACS enrichment technologies to do that, but it is quite a challenge. It isn't Cobra's expertise, and we pass that on for further manufacturing at that point.
Thank you.